I'm assuming everyone here is familiar with the terms cellular automata and the game of life. There's some details about both of those terms listed on this slide. In short, we're trying to develop initial conditions which produce the most complex behavior. Initial conditions are one of the two things that we can change about the cellular automata and analyze the results, the other being the rules, which we are keeping constant. A genetic algorithm was used as the optimizer for finding the most complex initial conditions for the game of life. A GA is a heuristic global search method that imitates the process of biological evolution. A GA has several components. It has a population, which contains chromosomes, and chromosomes, which contain genes. Of course, the GA requires a quantifiable metric of complexity that's ideally a high-level mathematical description. A multi-population GA was implemented for use in this research. This type of GA has multiple independent populations which communicate only occasionally. The goal is to maintain genetic diversity in complex solution spaces. The operators for crossover and mutation remain the same, but we've also included an operation called migration, where there's a chance for one chromosome to move to a new population at the end of each generation in the GA. Populations also change size over time. Fitter populations increase in size, and less fit populations decrease in size. The simulation is set up so that the initial conditions occupy a grid with a width and height half of the total board. The complexity is evaluated at time steps 40 to 60 so that there's enough time for the simulation to stabilize. For the chromosome encoding for the GA, we use direct encoding. We have a square grid of alive and dead cells that are reshaped to a one-dimensional string of ones and zeros.
Our first fitness function is fractal fitness, which is based on fractal dimension. Fractal dimension is predictive of developmental level and subjective rankings of complexity in plants. It's also a correlate of subjective beauty as ranked by humans, especially for abstract and natural beauty, which are the types of beauty we might hope to achieve from Game of Life. We use the box counting technique to measure fractal dimension, and from this calculated a normalized fitness score based on the deviation of the fractal dimension from the range 1.58 to 1.9. Then for all time steps, we average this fitness value. As just a reminder, fractal dimension is the log of the number of boxes it takes to cover a figure at a large scale over the number of boxes it takes to cover the figure at the small scale over the log of the magnification between the scales. So our large scale was a unit size of 8 cells, and our small scale was unit size of 1 cell. The reason we chose the range 1.58 to 1.9 is because many interesting natural processes, including random walks, diffusion limited aggregation clusters, and 2D percolation clusters, have similar fractal dimensions. And the precise range is not of critical importance because most random Game of Life runs have low fractal dimension, and this corresponds to low fractal fitness. So really any improvement is going to do something significant. Our next fitness function was cluster distance fitness. We found cluster distance from the center of the Game of Life board to be an interesting metric to improve because outward growth is a property of many natural systems. Circle formation is also relevant because cluster distance turns each cluster into a sort of agent that wants to be as far away from the center as possible. Combined with wanting to be as far away from the center as possible, Game of Life cells need a certain density to live. That is a distance from their neighbors. So this may result in circle formation via regularly spaced clusters avoiding the center of the board. We defined a cluster as a group of contiguous live cells using the Moore neighborhood, so 8 neighbors surrounding a cell. Distance was computed from the mean position of all cluster members, and fitness at one time step was the sum of all distances to the center of clusters greater than or equal to size k. We used k equals 6. And the overall fitness was the mean fitness of all time steps. Finally, our last fitness function is cluster size fitness. Cluster size is an interesting parameter to optimize because of percolation. Larger clusters have a higher chance of percolating throughout the entire system, in this case, from one end of the board to the other. The larger the clusters, the higher the probability a cell is in the largest cluster. Dynamic activity in the Game of Life occurs in clusters, and the larger and more connected clusters are, the more percolation of clusters we can expect, and the larger the emergent growing and changing patterns will be. Greater percolation means more interesting dynamic growth and change at a potentially larger scale involving more cells. So we could see larger patterns than normal wash across the entire board. Cluster size fitness is defined rather simply as the size, the cardinality, of only the largest cluster at any given time step. The overall fitness for a chromosome is the mean largest cluster size at all time steps.
So on the next few slides, we're going to take a look at some of the examples of the solutions that our genetic algorithm was able to find for each of the fitness functions. The first slide, we're going to talk about the fractal fitness function, fractal dimension. So we're trying to maximize the fractal dimension of the evolution between 40 and 60 time steps. In the upper row, you can see the results that we got for two different board sizes, and the two pictures that we have on the left-hand side, they have features that are stable for the game of life rules, and these features were present in the current form throughout the entire 40 to 60 time steps interval. And this was a little disappointing. We were hoping to see something a little bit more interesting, even on the smaller board. You can see the larger board produced more sophisticated outcome right away. But for the smaller board, for all the runs and all different combinations of parameters that we tried for the genetic algorithm, it was all coming out like this. So in order to fix this somehow, we introduced a different fitness function, which was a product of the fractal dimension and the density of the solution. And that function produced pictures that are present in the lower row of the slide. On this slide, we see examples that we got for the cluster distance function. And since the smallest cluster size that we allowed was 6, we expected to see lots of clusters of the minimal size of size 6, ideally situated around the border of the lattice as far from the center as possible. So in the top row, we see 3 different examples with different parameter variations for the genetic algorithm, and they all do produce similar patterns. They all have these leaf-like structures, which are 6 clusters of size 6. And they are situated towards the end of the lattice. So what's interesting is that the solutions that we found seem to be not only targeted towards maximizes the fitness function, but they also maximize fitness function within the time interval that we take measurements between 40 and 60 time steps. These leaf-like structures start to occur at around time interval 40, and then they persist throughout until time step 60, and they slowly start to dissipate when they start to interfere with the rest of the life cells on the lattice.
We found that cluster distance fitness had success producing circle-like shapes. On the right is the density distribution produced by cluster distance fitness, and on the left is the density distribution produced by random initial conditions. As you can see, it's almost an inversion.
And finally, on this slide, we have results that we got for the last fitness function that we used, the one that maximized the size of the largest cluster present. So in the top row, you can see you can see 4 screenshots from a time evolution of a single solution that we found. 43, 48, 55, and 60 time steps is the same solution. So at the start, we can see one large cluster, and then it slowly breaks apart as the evolution progresses.
We found that cluster size fitness significantly increased the largest cluster size, almost doubling the mean largest cluster size that we observed. However, it had little effect on the mean cluster size for all clusters.
We also performed an initial condition density analysis to analyze the effect that the percentage of cells alive in an initialized GA population had on a fitness function. We ran 3 trainings with each fitness function on a board size of 32 by 32, and we found that cluster distance had the only significant positive correlation as seen in the upper graph. Fractal fitness, as seen in the lower graph, had no correlation, and the results varied across each test run greatly. So, just a brief summary of our project. We used a multi-population genetic algorithm to optimize game-of-life cellular automata initial conditions to find complex solutions. The genetic algorithm fitness functions used were fractal, cluster distance, and cluster size fitness. We found that the GA finds initial conditions that result in simulations that achieve high fitness values in accordance with each fitness function.